Hey, thanks for our viewers joining us here with Contagion. I'm Grant Gallagher, and I'm sitting down with Dr. Jason Kindrachuk, and we're going to be talking about a lot of different issues to do with the microbiology of SARS-CoV-2, and we'll just dive right into it. Dr. Kindrachuk, what is your background with coronaviruses, and how has it impacted how you've thought about acting on this pandemic?
Yeah, so my background is really predominantly emerging viruses as a whole, and trying to understand the molecular pathogenesis as well as the circulation and spillover of these viruses. My kind of first introduction into the coronas was really with MERS. So 2013, 2014, when we started to see it spreading across the Middle East. We jumped in, with kind of both feet to understand what this virus was and whether or not there were any countermeasures that we could identify very quickly to fight against this. So since then, I continue to work on all emerging viruses, but that includes things as straightforward as outbreak and pandemic preparedness. So when SARS-CoV-2 emerged, that fit perfectly into my wheelhouse.
Great. And I saw something on Twitter that you were involved in coordinating supplies. Could you just briefly touch on what that's entailed?
Yeah. So we know here, at least in Canada, and obviously you've been impacted in the US, there are shortages on supplies because we have a lot of people going to hospitals and healthcare networks all at the same time. So things as basic as gloves, surgical masks, N95s, infrared thermometers. But we didn't really see a really, kind of a strong coordinated effort to do this either at the provincial or regional levels in Canada. So myself and a few other people here at the University of Manitoba basically created a network to be able to try and identify and distribute supplies, both locally as well as regionally. So we're kind of in the middle of that. We haven't quite had the case tolls of other regions in Canada or the US, but we see cases are increasing. So I think we want to be as prepared as possible.
Great. Okay. So could you start us off by comparing and contrasting the biology of this with the closely related SARS-CoV-1 and other coronaviruses like MERS?
Yeah, so I think we're starting to get a better idea of what this virus is, right? Keeping in mind that we're still 3 months out from when this really first emerged, or at least publicly was made made notable that it had emerged. What we understand now from the standpoint of sequencing, this virus is somewhat close to SARS coronavirus, to the original SARS-CoV. So in terms of looking at its phylogeny, it shares some similarities to that. But also based on sequence analysis, what we've been able to essentially surmise is that the likelihood is that this virus, much like SARS and MERS, or was postulated for those, it likely spilled over from bats. So we know that, again, it's a respiratory virus. It attacks the cells in the respiratory tract. It looks for the ACE2 receptor, much like SARS-CoV does. So that at least gives us a little bit of an idea of where we can look for potential novel therapeutics. But we know as well that what we're seeing is that it spreads very, very quickly. And I think as compared to SARS, what we're seeing is that the spread of this virus has far exceeded that, and we're now in the midst of a pandemic.
Absolutely. I mean, SARS certainly caused some suffering in clusters of outbreaks, including in your native Canada. There was that, I think was it Toronto hospital?
It was Toronto.
That had that tragic kind of blow up there, but overall, not the kind of spread that we saw with this.
No.
Do you have any sense of why that is? I mean, I know that this has a lot of asymptomatic carriers, for example.
Yeah, and I think that's a large part of it, right? Listen, we're still learning about the transmissibility of this virus. I think that hindsight is going to be 2020 when we finally go back and start to do some serology and understand how much of the population was actually infected, as opposed to those that were maybe confirmed or deemed probable based on diagnostics. But I think a lot of it is just the fact that it seems to be quite transmissible. But we have this large contingent of patients that appear to be able to spread the virus even in the pre-symptomatic phase, And that really makes it difficult for us to trace and understand where this virus is going to. So from a containment aspect, it's just been monstrously difficult to get this thing under control.
There was some concern actually about the heightened risk to doctors involving the chance of a kind of like a density of viral exposure in a
Yep.
way that is considered similar to dynamics with HIV.
Hmm.
Um, and I mean, that makes me think to something like the Wuhan quarantine, where you did have, familial spread and things like that, and the concentration and that burden on the health system.
Hmm.
Do you have any sense of whether that kind of uh, increased viral dose, I guess, is going to be impacting? I'm not sure if it would be the severity of infections or the likelihood of it being infected.
Yeah, so it's a great question. Actually, there was I was on a Twitter thread discussion about this last night with a couple of other PhDs. We've what we've basically seen is that certain viruses, basically have this kind of correlation between the amount of viral inoculum that is found during the course of infection as compared to overall disease severity. And we saw some of this in SARS with healthcare workers that were working with patients in very close proximity, in particular, patients that needed to be intubated or people that needed to do bronchoscopy. If you're right in basically close proximity, you have the potential or possibility that somebody could release, first of all, small particle aerosols that could be picked up in a short distance, but also a high viral load because you're right at the source. I don't think the data has really come out yet from COVID-19 to say, what that link is for healthcare workers. But I think certainly we're watching, and it is still, basically a testament back to this idea of why we really need personal protective equipment for our frontline healthcare workers. Because even if it's just a risk at this point, we want to try and minimize, the severity of disease in those folks as much as possible.